Hello, everyone, and welcome to today's webcast. I am Cheryl Rogers, the Director of Marketing here at Golden Helix. Our webcast today is covering prediction and meta-analysis, and is presented by Greta Lindsay-Peterson, our Director of Product Management and Quality. Greta, I'll hand it over to you.
Thank you, Cheryl. If you have any questions during today's presentation, please feel free to enter them at any time in the GoToMeeting questions pane. And one of the questions that is always asked is whether or not the webcast will be recorded and available, and the answer is yes. We usually try to get those up the same week. So stay tuned on that, and we'll talk more about that at the end of the webcast. For those of you who may not be very familiar with Golden Helix, we were founded in 1998, and we have been developing products for quite some time. Before SVS, we had a product called Helix Tree, which featured predictions with trees and random forests of trees. And we are now coming full circle with our SVS product and bringing some of that prediction capabilities back into our software, and I'll talk a little bit about that today. One of the other products that we have, if you're not familiar, is Genome Browse. Now, we do have Genome Browse in SVS, but we also have Genome Browse as a free standalone product, and we will be looking at Genome Browse as it is embedded into SVS a little bit today. Our newest product is VARSeq, and that focuses on clinical DNA sequencing workflows. And we do have numerous webcasts and resources available on VARSeq if you wish to learn more. However, this is not going to be the focus of today's webcast. As I said, we are going to be talking about our SNP and Variation Suite, or SVS, and this was created specifically for genetic research, and it was initially developed in the GWAS era. The features that I'm going to be talking about today are GWAS features, so GWAS is not dead. And with SVS, the performance and usability and visualization have made the software an obvious and popular choice for genetic researchers researching both GWAS and DNA sequencing, and has resulted in hundreds of citations in peer-reviewed articles. And we continue to advance our capabilities in both the GWAS and the DNA sequencing worlds, and we try to adopt the best practices methods and bring those into SVS to make sure that our researchers and users have the best and most widely used tools today. So just our approximate agenda. First, before we can talk about genomic prediction by itself, we need to talk about k-fold cross-validation that uses GBLUP, Genomic Best Linear Unbiased Predictors, and Bayesian Pi. This in and of itself can also be a genomic prediction method, but we will be talking about the two different ways to do genomic prediction. And then the last portion we'll be talking about meta-analysis. Now, these are not necessarily interconnected, but they're just two new exciting features that we want to talk about, and then we'll wrap up with questions and answers. We do have lots of genomic prediction resources already available that talk about different aspects of what we have available. And if you're interested in learning more about genomic prediction, or a little bit deeper into the methods, I encourage you to check out these resources. Once we do have the slides available online, you can click on the pictures to jump out to either the webcast or the blog post listed here. As I said, we have numerous genomic prediction resources available, so I will just provide a quick overview today of what we mean when we talk about genomic prediction and the methods available. Genomic prediction is used to take a set of loci or markers and a relationship matrix and to be used as random effects, and builds a mixed linear regression model. If the phenotype or the Y value is known, this gives you the contribution of each loci to the model, and this can identify the markers most associated with the phenotype. If the phenotype is not known for a group of samples, you can also use the allele substitution effects for each marker and the known phenotypes to predict the missing phenotypes. Now, there is known shrinkage in the prediction phenotypes, but it is a uniform shrinkage as long as your phenotype is fairly normally distributed. And SVS will take a non-normal distributed phenotype and normalizes it to make it more easily predicted. SVS currently has 3 genomic prediction methods available. GBLUP, which assumes that all the loci contribute to the phenotype. Bayesian, which estimates the effects of the gene loci together with the parameters required to define the probability distribution over the events. And it assumes that the prior probability that any SNP will have no effect is fixed. By default, we fix this at 50%. And then Bayesian pi will also allow that prior probability for the SNP to vary. The data that I'm going to be using today for the genomic prediction portion is some data that we have used in the past from the bovine HapMap project with the simulated phenotype. From the data set of 402 Bos Taurus cattle, I randomly held out 51 samples from 5 breeds. We will use the remaining 351 samples to build a genetic model and apply the model to the holdout set to predict the phenotype for these samples. Now, since we have a phenotype for the holdout set, we can also take a look at how well the predicted phenotype compares to the simulated phenotype. To build the model, we will use k-fold cross-validation. You may have seen this diagram recently. We have used this to illustrate in previous webcasts and blog posts how a 5-fold cross-validation process works. With each fold, 1/5 of the data, in this case, since it's a 5-fold cross-validation, has the phenotype set to missing, and the remainder, the 4/5 of the data, builds the model and predicts the phenotype for the holdout set. And then we do this 5 times and then take the predicted phenotypes for each 5th and combine that together, as well as compute some prediction statistics. Each sample must have a phenotype value to be included in the k-fold cross-validation. If you already have your samples that are missing phenotype data, you can, and they're already in the same spreadsheet, instead of doing a k-fold cross-validation, you can just run a single GBLUP or Bayesian pi and use the known phenotypes to predict the missing phenotypes. Or if they're in a completely different set of samples, which we're going to talk about today, you can use the k-fold to create a model that can be applied to the set that's missing the phenotypes. You can also include covariates to build the model, but the resulting model that's going to be used to predict the phenotypes for a new set of genotypes must have the same covariates present for the new samples. And that may be a restriction that we can lift in the future, but right now we do have this restriction. With the latest release of SVS last week, we now have the ability to run k-fold cross-validation multiple times with just specifying your parameters once and setting the number of folds and the number of iterations. Set all your parameters, click OK, and it'll run iteratively through the process. And this will allow you to compile iteration statistics that can help inform the ability of the genotypes to predict the phenotype. With the binary phenotype, you do get, with the iteration statistics, a measure of sensitivity and specificity, and with quantitative phenotypes, you do get correlation statistics. The results from running GBLUP with 5-fold cross-validation on the 351 samples and 20 iterations can be seen here. And because the folds vary, each time you run this, you might get slightly different results. In this particular case, you can see the correlation, or the R-squared values here, and the blue line is the mean R-squared value with the confidence band around it. And in this particular case, the 16th iteration resulted in the highest R-squared value. So I went ahead and plotted the predicted phenotypes versus the phenotype for that iteration 16 and added the correlation trend, the regression line between those two phenotype values. And again, the R-squared value was 0.258. I also colored the phenotypes by, or the samples by the fold number. As you can see, the folds are pretty well distributed across all the phenotypes. So let's go ahead and set that up in an SVS demonstration. So bring up SVS, and I'm going to start with my cross-validation starter project. And in this project, I do have the animals for building the model, as well as the animals for prediction. In this particular demonstration, we're going to ignore the second spreadsheet and just work with the first spreadsheet. And let me make this a little bit bigger. We see we have some phenotype values, 351 samples, and my genotypes are over here. If I expand this a little bit more, I can see that I have my marker map with chromosome and position in it, and some Illumina strand information as well. The first thing that I want to do is make sure that I filter the genotypes down to good quality markers. So I will just perform a quick filtering by marker statistics using the defaults for those top three, and we'll run this. And with this size of data, it runs pretty quickly. And this is the filtering results spreadsheet. I'm going to go ahead and close that, and we'll create a column subset spreadsheet. And let's go ahead and rename this to quality filtered at the end. And from here, we are we'll also do LD pruning to reduce the number of markers that we have to search over. And this is going to take just a little bit longer, not too much. And while this is running, today we're going to be using GBLUP with k-fold cross-validation. You can run GBLUP, Bayesian, BayesiPy, or set up the parameters all at once, and it'll run sequentially. Bayesian and BayesiPy do take a little bit longer to run, and it is not necessary to include every single marker that is good quality into the set that goes into running Bayesian and BayesiPy, as it's looking for the loci that have the most effect on the phenotype. So we do also recommend that you further reduce the number of markers that you're looking at with the Bayesian methods, perhaps by running a simple correlation trend test to pick the top % of markers, and then just use that to make it run faster. We have had people running it on their full set, and it can take, if you have a couple million markers, it can take 3 days to run on 2,000 samples. So just a fair warning on that. All right, so now we've LD pruned our data. We'll create another subset here and rename this LD pruned. Okay, and then we'll close this other two spreadsheets because we're going to use this one here. And again, we are starting with genotypes, but what we really need to do is sorry, let me back up. We'll go ahead and set our phenotype here. And what I'm going to do is I'm going to launch the k-fold cross-validation dialog. As it's launching the dialog, what it's actually doing is recoding the genotypes into an additive model, 0, 1, and 2, where 0, it means no copies of the minor allele, and 2 means 2 copies of the minor allele. And by doing this first, it makes sure that any fold will have the same exact numeric coding, because depending on how you pick your fold, your minor allele could potentially change if the frequencies were pretty close to 50% to begin with. Now that we have that recoded, I'm actually going to cancel this and then close this spreadsheet. And as you can see here, we've got the recoded spreadsheet. And I'm going to run the genomic, or create the genomic relationship matrix from this spreadsheet so that we can reuse the same exact spreadsheet every single time. If we don't create this in advance, it will create it when you run k-fold, but it'll create it for every iteration. And I want to save a little bit of time today, so we will create this first. And this is just, the relatedness between samples, and we can see, these two samples are again, it's the same sample. It's a value of higher than 1. This is not a true identity by state matrix, so your values can be more than one. It's just a gradient on how much effect each allele has and in how related they are. Okay, so now back to this spreadsheet that's numerically recoded. We'll launch the k-fold cross-validation again. Again, I'm going to run genomic best linear unbiased predictors, or GBLUP. And for the sake of demonstration purposes, I'm just going to run 3 folds in one iteration. And I do want to use that pre-computed genomic relationship matrix, so I'll select that. You can correct for additional covariates, and I'm also going to stratify my folds by breed to make sure that I get a good representation in each of those folds, and we'll go ahead and say OK. And while this is running, it's going to first randomly pick which fold each sample is going to go into, and then run GBLUP for that. So we're just going to let this run, and you can see it's working very quickly. And it should almost be done. After this is finished, we'll take a quick peek, and then we'll pop over to a project where I have finished, and where I've run 3 folds and 3 iterations. We'll take a peek at that, and then we'll be able to look at the iteration statistics from that spreadsheet. All right. So the final results from the k-fold, again, results in, this is the original phenotype that we specified. The predicted phenotype, again, this was the phenotype from the fold, and it tells you that this phenotype was predicted in fold 3. And from here, we can actually, let's go back to our project here real quick. So we can see we've got results for fold 1, 2, and 3. And in each fold, we have the samples used. So in this particular fold here, if we scroll over, you can reuse this spreadsheet. You can see that some of the values are set to missing. And then we get the allele substitution effects for each fold, as well as the fixed effects coefficients, which is just the intercept. In this case, since we didn't have any other covariates, and the estimates by sample for each fold. And then this information is carried forward to that final results spreadsheet. The final results does give you the predicted phenotype, as well as the allele substitution effects, fixed effects coefficients, and the summary statistics. The allele substitution effects are the average of all the allele substitution effects across all the folds. And then the other statistics are computed from that. With our final results, let's go ahead and we'll do a scatter plot here. Again, we'll do predicted phenotype on the x-axis and the phenotype on the y. And this will create a scatter plot. Let's get rid of that. And now we can color this by the fold predicted in. And we can add a regression line if we want, R-squared information, and so on. But we can see in those 3 folds, it's very well distributed across all the phenotype values. Now let's pop over to, discard this, the finished project. And here I've run 3 iterations, iteration 1, 2, and 3, and I've run this twice. The first time I ran it with the quantitative phenotype, and then the second time I ran it with a binary phenotype. So just to take a look at the iteration summary statistics. For the quantitative phenotype, I do get the coefficient, Pearson's coefficient, R-squared, root mean square error, and so on. For the binary phenotype, I also get area under the curve, Matthew's coefficient, accuracy, sensitivity, specificity. So you can create an ROC plot from, or curve from this if you ran it multiple times. And if we were to look at, to go back to our quantitative results, we can look and see which one has the highest R-squared value. In this case, it's iteration 3, so we could then carry forward and use the results from iteration 3 to predict our data, but we're actually going to use results that have 20 iterations for the next step. So let's go ahead and get back. Let's close this out and get back to our presentation. In agricultural contexts, as well as for researchers working with genomic human data, if you have genotype data for more samples than you have phenotype data for, you may want to estimate the phenotypes for those samples missing the data. Or perhaps you collect data in chunks year after year and want to know how predictions change. With genomic prediction, you can build a model once on the data that you have and then test it again against the new data and the genotypes that you may or may not have phenotype data for, and use that to either build a new model or test to see how well the model is still fitting, and then you can continue to build up the model or refine the model, or you can just reuse the same model time after time. Once we have the results of k-fold cross-validation in particular, we need the allele substitution effects, how much each marker contributes to the model, and the fixed effect coefficients, and minimally the intercept coefficient, and then we can apply the model to a new data set. To ensure we're comparing the same effect alleles, the data is first recoded to numeric, like in k-fold cross-validation, and the major and minor alleles are compared. If the strands need to be flipped, they are, and if the alleles are ambiguous, then the marker is just excluded from the prediction. The results from genomic prediction is just simply a spreadsheet of predicted phenotypes. We can then join the predicted phenotypes to the simulated phenotypes to see how good the prediction model was, or just take that data and move forward in making decisions however your research takes you. So let's jump to the genomic prediction demonstration. So open our prediction starter project, and here again I've got 20 iterations using that same quantitative phenotype. And one thing that I didn't show is that after the data is recoded into a numeric model, the minor allele and the major allele are added to the marker map, so this information will be used moving forward as we're comparing strands. If that information is already in your marker map, it will prefer the information that you already have rather than inferring it from your data. So if you want to override the alleles used, you can use the information from the marker map if you have it available. All right, so we've already run k-fold cross-validation. Now we need to decide which iteration we want to use to predict our new genotypes. So again, I'm going to prefer the R-squared value, and I can sort this in descending order. And by doing that, I can see that iteration 15 has an R-squared value of 0.276. So we will go ahead and use iteration 15. As you can see, I already have this marked blue. So let me just expand that. Again, with iteration 15, I have the allele substitution effects, the fixed effect coefficients, and the summary statistics for this iteration. And I did run 5 folds in this particular case, and so you can see how well this is fitting. You can also see that some folds had a better fit than other folds. In particular, fold 5 had an R-squared value of 0.3, whereas fold 4 had an R-squared value of 0.17, but altogether this was the best fit of the predicted phenotypes to the known phenotypes. All right, so let's open up our holdout set, the 51 samples. As you can see, I've got, again, my phenotype values, but we're going to ignore that and pretend we don't have it, and our genotype values. And you can see here that before I had 39,000 markers that I used, and I have all of the markers from the 50K array in the spreadsheet. I don't have to worry about that. It's only going to use the markers that it has allele substitution values for. So from here, I'm just going to go to Genotype, predict phenotypes from existing results. And again, that slight delay was recoding the spreadsheet and or getting the spreadsheet ready. It's already set up to use defaults for GBLUP. If you had Bayesian results, we go ahead and recommend that you change it to not centering the data and removing homozygous markers. But again, since I have GBLUP data, I will use the defaults. And all I need to specify are the allele substitution effects. I'm just going to scroll down to the iteration 15 results. And you can include additional covariates, but again, you need to have the same covariates from both your prediction and your this particular data set. And since I only have a subset of breeds, I could use breed here, but since I only have 5 out of the 16 breeds, I don't want to do that as I won't have all the information needed. You could also transform, specify that you had data that was transformed, and then you'd enter that information there as well. Just click OK. And this matching alleles portion just takes a second. It's making sure that there aren't any the need to have their strands flipped. And after it's done with this, applying the model happens in the blink of an eye. And as you can see, it's already creating the results. And there we go. So there's our predicted phenotypes. From this, we'll go ahead and join it to the spreadsheet that I have behind. So I can just take a look at the results. I'll just join it to the original one. And I'm going to run some numeric statistics. So I'm just going to go ahead and inactivate these so that it doesn't clutter the results. We'll set the predicted phenotypes to dependent. Let's just take a look to see how good that prediction did. Run a correlation trend test and linear regression test. And in this particular case, we have a correlation trend R of 0.47. If I want to see what that converts to in an R-squared value, I'm just going to copy this cell and pop back to the project navigator. We do have Python scripting available in SVS, but more than that, we have a Python shell that allows you to do quick calculations. So I'm just going to Control-V paste that and **2 to square it, and I see that I get an R-squared value of 0.22. And then if we go back to our initial results here that are joined together, I can just right-click on this phenotype 5 and plot an XY scatter plot. And this time, instead of coloring it on fold, I'm actually going to color it on breed, and we'll have the 5 breeds that are in this set. And you can see here, let's make the number, the size here a little bit bigger. We do have breeds that are clustering together. In general, the Beefmaster and the Limousine are clustering down here, whereas the Angus and Red Angus are clustering up here, and the Hereford are all through the middle. And that's, just goes to show there is some breed bias here in this prediction, but maybe that is well warranted. Maybe the phenotype predicts some measure that differentiates these breeds. All right, let's see. You can also add a regression line based on the regression intercept and slope here, but I will leave that to be discussed at a later time. So let's go ahead and exit out of this, discard our changes, and get back to our presentation. So that was a quick overview of K-fold cross-validation and genomic prediction in SVS. Again, we technically have 2 different types of genomic predictions. If all of your data is in one spreadsheet and some of your data is missing, for your phenotype, you can run a single GBLUP or Bayes-C/Pi to predict those missing phenotypes. Or you can run, if you have all of the phenotypes known for a certain set, you can run K-fold on that set and then apply the prediction model to the set that's missing the phenotypes, and that's the second way of running genomic prediction. The next thing we're going to talk about today is meta-analysis. Our most recent customer survey that perhaps you saw earlier this year and participated in, meta-analysis is one of the most requested features. And we're pleased to announce that coming next month, we will have meta-analysis in SVS, and I will give you a preview of this feature today. Meta-analysis can be used to test the effects of markers across multiple published studies or population groups within the same study, and it can be useful when you do not have access to the raw data, as well as when you do, if you want to use meta-analysis to compare groups such as population groups. And this forest plot that I have here was created in Excel, but I created it in Excel using data that I was able to get out of SVS. With meta-analysis, if you're not familiar with it, you will have to make a few choices. The first choice will be which studies to include in your analysis. Obviously, you want to pick the studies that had rigorous quality control and data cleanup and that match as closely as possible. Some flexibility is allowed in which data that you use for the meta-analysis. However, you have to have the same basic types of results that you use. To use studies that report p-values, you will need also a measure of the effect direction and the effective number of samples. For case control studies, this means that you will either need the breakdown of number of cases and controls per marker, or at a minimum the number of cases and controls per study. For quantitative phenotypes, you will either need to know the total number of samples per marker or the total number of samples over the whole study. To use inverse variance methods, you will either need odds ratio and its confidence interval, or effect size and its standard error. And this table right here shows that we've got 5 studies. What do the studies have? Which group of meta-analysis should we use? P-values or inverse variance. And each group has 4 studies that have information in both, but study B only has the effect direction. It doesn't have any p-values, so we would have to exclude that. And study E, if we look at the inverse variance based input, study C doesn't have any of that output, but the rest of the 4 studies do. So in this case, I would choose to use either the odds ratio or the effect size inverse variance based method. There are also two main types of meta-analysis. There's a fixed effect meta-analysis and random effects meta-analysis, and each type has certain assumptions which we will quickly go over. The fixed effects meta-analysis is, as most fixed effects methods are, the most naive. It weights results based either on the square root of the sample size or the inverse variance of the standard error of the effect estimate. It also assumes that all of the studies are based on the same population and comprise the whole population of studies and are predicting the same phenotype. This in general is not very realistic, but there is a genomic control option that can be added to a fixed effect meta-analysis that can help with some of the violations of the assumptions. And here's a table of statistics that are used in fixed effect models. I'm not going to go into detail of these statistics, but you can look at this paper to get more information on these statistics. And it'll also be available in our manual when we have this ready to ship in our software. The random effects meta-analysis method assumes that all of the studies chosen for the analysis are randomly chosen from all the studies and that the effects vary around an overall average effect. The weights for this model include within study variability, or the random error, and between study variability, also known as heterogeneity. And I apologize that there are numerous statistics that you need for random effects, so those are pretty small. Again, this will be in our manual or on the slides online if you want to dig into that further, or you can also get them from these links here. Meta-analysis in SVS allows you to pick either the inverse variance approach or the sample size approach. Both the fixed and random effects models are computed, and there is the option to compute the genomic control fixed effect model. Once you set the parameters for the first model, the options will be restricted for subsequent studies to guide you in choosing valid options. So here is the dialog for the options for the first study. If you decide to go with the sample size based inputs, you would select that here, and then you would only see this portion of the dialog for subsequent studies. If you went with the inverse variance based input for the first study, then you'd only see this portion for the subsequent studies. There is a dialog that comes previous to this, which we will see when we move into the demonstration portion, where you select all the spreadsheets that you want to use that contain the results from the studies. The output that we get out of meta-analysis, again, includes the fixed effect model and output and the random effects model output, which is the same as that for the fixed effects. We also get Cochrane's Q, I-squared, tau-squared, and optionally genomic control variants of the fixed effect model. And here is a plot in Genome Browse of some meta-analysis results. This is the meta-analysis results, and the red line is a genome-wide Bonferroni correction significance level. And as you can see, these are all the individual p-values per population. None of them rise to significance, although some of them are more significant. However, when we do a meta-analysis on these, then we have quite a few markers that do rise to significance in this area. All right, so let's go ahead and take a look at that in SVS. Let me adjust this a little bit here. Okay, so I've already, I didn't introduce the data. I'm just using HapMap data. This is on the older NCBI 36 array, or sorry, genome coordinates, but it will serve our purpose today. I did go ahead and do the same quality filters that I did with the bovine data. I also LD pruned it. I'm only looking at chromosome 4 just to isolate and the results. Meta-analysis does not take very long to run, but this will help us just focus on one area. And after I did the quality control filtering, chromosome down to chromosome 4 and LD pruning, I went ahead and recoded this into a numeric additive model, 0, 1, and 2. And again, that adds the major and minor allele into the spreadsheet, which ensures that I'm working with the same strands. And then after that, I split this particular data set on population. So here, if we look at all the population, look at the pie chart, let's see how. We've got numerous populations here, 11 populations. So I split that into 11 spreadsheets and ran an association test on each of these. And we'll do a meta-analysis to combine all of these results to see if there are any markers or loci that trend the same way across all the populations. So the association test results that I did, I used a binary phenotype. So for this particular population, we had 145 false, did not have the phenotype, and 22 controls, or cases, sorry, 145 controls, 22 cases. And so I was able to run an association test that gives me everything that I could want. It gives me p-values, regression p-values. It also gives me regression odds ratio and the confidence bounds. So I not only have the p-value results, but I also have the effect size and odds ratio options. But today we'll use and focus on the odds ratio option for meta-analysis. So from the Tools menu, since I'm going to be selecting multiple spreadsheets, I'll go to Perform Meta-Analysis. And I'm going to go ahead and select all 11 of those spreadsheets. I'm just going to go ahead and leave the default options, but you could further restrict this to say that you needed to have 9 out of the 11 studies have a valid result in order to include the marker in the meta-analysis. We'll just leave that at 2. And again, here's the dialog for that first study. It's picking the row labels as the marker names, which is great. And I'm going to go down here to the odds ratio input. And so I'm going to just quickly search odds ratio. And now I need the odds ratio lower bound and the odds ratio upper bound. And I'm going to click next. And here's for the second spreadsheet. Now I have modified this script to make it easier for the purposes of this webcast, so it'll automatically pick up those columns that I want. I just need to select that option. When we ship, you may have to actually select that column yourself. So I'm just going to quickly go through this. And one more. And meta-analysis is that quick. So with my output, I have it in groups. So first I have the fixed effects output, this overall p-values for the fixed effects. Next I have the RE analysis p-value, the random effects analysis p-value, and its negative log 10 p-value. The number of studies for which the marker was valid, overall effect size and standard error, and these are for the fixed effects model. And then I also have the random effect size and its standard error and its z. I also have the Cochrane's Q, I squared, and tau squared. Then I have all of these study results that I actually used to build the statistics for the meta-analysis. So we can use these later to create a plot. The first thing that I want to do from this results is plot the negative log 10 random effects p-value. So I'm just going to right click on this column, plot variable in genome browse. And again, it's the NCBI 36. We do see a nice peak here, and I'm going to just zoom into chromosome 4 and then further zoom into that peak. Okay, let's just click on this marker here. And I'm going to copy that RSID so that we can jump to it in just a second. Now, close this. From this spreadsheet, back to the spreadsheet, I want to plot all of the regression odds ratios. So I'm just going to put in a filter odds ratio, and I can see showing 11, which I had 11 studies. Click all of them, and I want all of them in one graph. And in order to jump to that RSID, I do need to add dbSNP 130. And then I'll paste in that RSID, and we'll zoom out a little bit. Let's put a marker mouse anchor right there. Okay, so it's kind of hard to see, how good these are. Let's change our zoom here a little bit. Let's change the Y range to just go to 10. And let's add a line at Y equals 1. Give it a little bit thicker width. So now we can see with this particular marker, we'll quickly add the p-values for that particular, for the meta-analysis. And we'll hide RSIDs. So you can see that for this particular marker, all of the odds ratios are above 1. They're all trending the same direction. For this particular marker, we have some odds ratios that are below 1, and as a result, the meta-analysis p-value is lower. And here, even though all of these are below 1, they're all trending the same direction. So again, the p-value for that combined meta-analysis is also high. So this is the power of meta-analysis. It can help you in doing a traditional meta-analysis where you're scraping results from geo or online databases that just give you the results. You can look at a single marker at a time if that's all you had. But in addition to that, you can use this as an alternative, which some researchers have recommended, to population correction, stratification, PCA correction. This is an alternative method if you analyze all of your population separately and then perform a meta-analysis. So let's go back and we'll just close this out, discard those changes. Oops. Let's go back up here. All right. So to summarize, SVS now has k-fold cross-validation, which can be used to build a genomic prediction model. And then that prediction model can now be applied to new data without having to worry about merging and rerunning and strand aligning. And then you can also run that k-fold cross-validation multiple iterations with just one setup of the dialog. You can start it, walk away. If you run Bayesian Pi, you can start it, walk away, and come back in a couple of weeks if you want to run a lot of iterations, and you'll have your results all nice and compiled. In the next month, SVS will also have meta-analysis methods available. While meta-analysis is available in other software packages, having it in SVS provides not only the convenience of our data import and manipulation tools, but also visualization and genome browse, and takes advantage of our user-friendly GUIs. One of the things that meta-analysis is also going to bring is that all of our standard GWAS statistics are now going to provide the input that you need in order to perform a meta-analysis. What does that mean? That means that we're going to make sure that we tell you either in the marker map or in the spreadsheet what the major allele was and the minor allele was that we used for the test. We'll also tell you the sample sizes. If it's a case control, we'll tell you how many cases were used for that marker, how many controls we used for that marker. If it's a quantitative phenotype, we'll just tell you the number of samples we used for that particular marker or test. We'll make sure that you have what you need in SVS moving forward to perform meta-analysis on all of your GWAS results. If you want to use your perform meta-analysis on results that you have from the past, we'll also have the ability to add that information to the marker map in retrospect, as long as you have the original data to know what the major and the minor allele is, and then you can use that going forward. So with that, if you have any general questions about Golden Helix or any of our software products, feel free to email info@goldenhelix.com. If you want to request an evaluation, that's available on our website as well. And if you haven't already done so, please go ahead and put in any questions that you have into the questions pane. And while you're doing that, I'll go ahead and turn it over to Cheryl.
Thank you, Greta. As Greta mentioned, we'll answer some questions. I just have a couple of quick things. The webcast recording and slides will be up on our website tomorrow, maybe today, because I'm not sure I can get that done. Our next webcast will be coming up on Wednesday, June the 10th. That will be presented by Bryce Christensen on Genotype services. And Bryce will present an example of how VARSI can be used in whole exome sequencing research. That's all I have, so we will take some questions.
All right, so I apologize. This was a very methods-heavy webcast. I am using a lot of I did use a lot of acronyms, mostly because this is, I didn't want to cover too much of the content that we already have on and webcasts available. So if you were a little bit lost, I do encourage you to go ahead and check out some of our other webcasts, especially with the genomic prediction information, we have all of this very well, thoroughly documented in our manual, and I encourage you to take a look at that as well. So with that, again, I apologize. The genomic prediction, it's not new. We've been continually adding to that since or for a few years now. And if you have any other suggestions on how we can further advance our genomic prediction capabilities, I would be more than happy to take a look at what your requests are. Our focus for the next half of the year is changing slightly, moving away from GWAS and giving some attention to other areas of our software, but I am always happy to take a look at other methods that are out there in other deficiencies that we have. So as far as meta-analysis, the meta-analysis that we implemented in our software is highly borrowed from METAL, M-E-T-A-L, and it's also used some other packages from the group that does MPute2, and I can't remember the name of the meta-analysis package off the top of my head. But we took all of those packages that run on the command line, we put them in our software, we combine them all so it takes the best from each of these packages and puts them in one place, and then makes it easy for you to just to use without having to know the command line options. And again, when we have this ready, we will make sure this is really thoroughly documented so that it's easy to figure out which options you want to use. And if you're not very versed in meta-analysis, it should be straightforward to get going on that. As far as that, Cheryl, do you see any other questions?
I did see one on there that we didn't quite cover. Can you use the software to look at whether two or more variants in combination produce a strong association?
So that would be more of like our collapsing methods, and so that we consider that more in the realm of DNA sequencing research. And so we do have the collapsing methods available. It does require knowing, what the alternate allele is. So so it requires having a reference allele and an alternate allele. Again, that's more in the collapsing methods. So yes, we have that. And we also have ways of, even if it's not a traditional DNA sequencing variants coming in from a VCF file, we can still aggregate information over genes. It's a little bit, if it's coming from a microarray, your probes are not necessarily in a gene, and so you might need to expand your definition of a gene region by a little bit, but we do have that capability. So we have ways of making this work. If you're trying to make it work for GWAS, it might take a little bit more manipulation to make sure that you're setting your study up properly first, and then we can move forward.
Okay, and I actually see one more. Will meta-analysis be included in the price of SBS, or is it an additional add-on tool?
All of everything that I showed today, it's included in the price. It's just an add-on that we're continuing to add on to our software. SVS, all of our updates are free. We don't generally add on charges unless it's something that we have to then also pay a third party for. So for instance, in SVS, we've got a base price, but if you want to add PBAT, then that's an additional charge simply because we have to pay for having access, having PBAT within our software. If we have any other features that we can add on at a later date that we have to also pay somebody for, then we would have an additional charge. But any of the methods that we add into our software, it comes with the standard charge.
Perfect, thank you. I think that's all for today, so we can go ahead and wrap. Greta, we certainly appreciate your time. We know you're very busy. And thank you out there to everyone for joining us. I will have the recording and slides up just as soon as I can. Enjoy your Wednesday.